The following program is part of a college course. These telecourses allow you to earn college credits at home. For more information, contact a college or university near you.
Nature, in all its wondrous and unusual forms, displays an extraordinary diversity of shapes, sizes, colors, and textures. Even within a particular species, there's a variety of distinct characteristics and traits. Yet upon closer inspection, it's possible to see similarities within types of life forms, likenesses that are far too remarkable to attribute to some random genetic accident. It's these special similarities that can only be explained by a pattern of continuity known as inheritance. And it's through these patterns of inheritance, through this delicate but deliberate passage of characteristics from generation to generation, that the cycles of life are assured. The search for the secrets of inheritance began in a monastery near the Austrian Alps. Gregor Mendel, an Augustinian monk, performed a series of experiments that would revolutionize the field of study that we know today as genetics.
Mendel's approach to looking at characteristics that were inherited is to look at a single characteristic at a time. And when he bred his peas, he was the first person to really do any statistical analysis and bring numbers into it, and really the first person to say, I'm going to look at individual characteristics apart from all the others, and follow that one separately.
Mendel's meticulous documentation became a blueprint for research into the hybridization of plants. Kirk Larson has become a modern-day Mendel in his quest to build the better strawberry, larger, sweeter, more resistant to disease.
In a lot of ways, what we're doing is just what Gregor Mendel did. Move pollen between two parents and then harvest the seed, plant the seed out, and look at the progeny. That's exactly what we do here in the Strawberry Breeding Project. Move all of the pollen.
Like the pea plants, strawberries self-pollinate. Each flower produces pollen on the anther, the male part of the flower, and releases it to the female part known as the stigma. In order to prevent this self-pollination, Dr. Larson removes the anthers, then selects a pollen-producing flower from another documented parent plant.
So in about 3-4 weeks from now, we would be harvesting this, what we call the fruit, to get the seeds. Each one of these little seeds, or achenes, is a fruit that contains a seed inside. And each one of these achenes is then genetically unique and will give a plant that is genetically unique.
The plants produced from these achenes, or seeds, are called the F1 generation. When Mendel observed his peas at this stage, he noted that one of the expressions of each trait had apparently disappeared.
When Mendel crossed two true breeding stocks, he found that all of the offspring, as a result of that cross, produced only one visual trait. He named the trait in that generation that was apparent a dominant trait, and he identified the other one that did not occur as a recessive trait.
Dr. Larson carefully observes the F1 generation, collecting data weekly throughout the growing season. He is then able to define which plants are desirable for the next phase of research.
From a population of 7,000 to 10,000 seedlings, we would evaluate that population and then select the best 200, and then allow the 200 to just propagate themselves right in place during the summertime. Following which, in October of the third year, we would then come in and dig up these clonal plants, the daughter plants, and take them to an evaluation field. It's at that point in the third year that we really begin to assess the full genetic potential of these items.
Through painstaking documentation, Dr. Larson can trace the plants producing the most desirable characteristics back to the parent cross. This information is recorded, and these plants are crossed to form the F2 generation as the process begins again.
Mendel's particles, which he called factors, are now known to be genes. These genes are on subcellular organelles that exist in the nucleus called chromosomes.
Genes that are responsible for different forms of a trait are called alleles.
If the alleles in an individual are absolutely identical, then we call this homozygous. You can have the alleles be the same and be dominant, or you can have the alleles be the same and be recessive. When you have two alleles in the same cell, i.e. in the same organism, but the alleles are different so that one is dominant and one is recessive, we have a situation known as heterozygous. When you cross two heterozygotes, this is called a monohybrid cross.
While pea plants have two alleles for each trait studied, this is not true for the strawberry plant.
The strawberry is an octoploid, meaning it has 8 sets of alleles that control expression of any given gene. And so there's a degree of complexity beyond a diploid where there are 2 sets of alleles, just 2 alleles that control expression of a gene.
Because Mendel's peas had just 2 alleles for each trait, he was better able to recognize the simple pattern he obtained from several monohybrid crosses.
A trait such as tallness is governed by 2 alleles that separate when male and female gametes are made. Each gamete gets 1 allele. To chart potential offspring, scientists use a device called a Punnett square. Male gametes are placed along the side of the square, with female gametes on top. We symbolize the dominant tall allele with a capital T, and the recessive short with a small t. Because each heterozygous parent has a dominant and recessive allele, they can produce 2 types of gametes. This is the basis for the law of segregation. Using the Punnett square, we can depict the genotype or genetic makeup of potential offspring. Genotypes determine the physical appearance or phenotype of the resulting plants. The dominant phenotype, tall, is expressed in 3 of the offspring. 1 is homozygous, 2 are heterozygous. The 4th possible offspring, being homozygous recessive, is short. This cross produces the same 3-to-1 ratio obtained by Mendel.
After documenting the results of single-trait inheritance, Mendel studied the inheritance of 2 traits at the same time.
When he did a cross of this sort, it became apparent from the data that he was collecting that these traveled independently of each other.
This observation of dihybrid or dual factorial crosses led to the development of Mendel's law of independent assortment.
A dihybrid cross involves parents heterozygous for 2 traits. Here, they are shown as P for the dominant purple color and T for dominant height. With no special pattern indicated, male gametes could be formed with the capital P allele aligning with either the capital or small t. The small p allele would align in a similar manner. The heterozygous female produces the same combinations. The offspring display 16 possible combinations. 9 are tall with purple flowers, the dominant expressions. 3 are dominant tall with recessive white. 3 more are recessive small with dominant purple. A final combination is homozygous recessive for both traits. Again, the 9:3 to 3:1 ratio, determined by independent assortment, mimics Mendel's observations.
Strawberry growers benefit from years of trial and error on the part of researchers attempting to create an ideal combination of traits within a single strain of plants. A new variety is born when a combination of traits is produced that is significantly different from what presently exists.
We can baby the plants and get high productivity out of them, but it's really out in a grower's field that we'll see the true fitness, the real expression of the genetic potential of that plant in the real world. Typically, that's about a 7-year process for us.
This kind of painstaking labor has resulted in over 20 new, more appealing varieties of strawberries over the past 50 years. But more important, Dr. Larson is proving that it's possible to use Mendel's techniques to identify equally significant patterns of inheritance. Since ancient times, the image of the white dove has symbolized the quest for peace. Official ceremonies worldwide celebrate this image as flocks of birds are released to swirl and soar over admiring crowds. Yet the birds they see may, in reality, be white homing pigeons, specially bred cousins of the dove.
Our business started with the 1984 Olympics, and the Olympic Committee contacted me and my wife and asked us to provide a flock of pure white homing pigeons for the opening and closing ceremonies. They've been used in releases for the launching of ships, and they just bring a special joy to those that see them.
These hybrid flocks are an example of one of many notable exceptions to Mendel's laws of inheritance. Because of the variety of color combinations seen in pigeons, many results are possible.
The most common color patterns in the homing pigeon is what we call ash red. It's a very beautiful color. Below ash red is what we call blue-black, and that's what we see in the common street pigeons, the ferial pigeons.
If these two birds are mated, which will dominate, the red or blue-black?
The heterozygous ash red is exemplified in this pigeon. As you can see, there are dark feathers and a dark beak on the pigeon, and this tells us that this bird is carrying blue-black, physically or phenotypically, but the gene type is heterozygous.
By expressing both traits, these birds demonstrate the Mendelian exception known as codominance.
Codominance is when both alleles express pretty much independently of each other. It's a term that we use when a gene can have 2 or more states which can be expressed together. And perhaps the simplest example is the ABO blood groups, where in humans there are 3 alleles of this particular gene: the allele capital A, the allele capital B, and the allele capital O. So you can be AB, and in that case, it is a case of codominance, because if you have an A copy and a B copy, you put both proteins on your blood cells, and you are defined as being in the AB blood group. Incomplete dominance is a different situation where the 2 alleles homozygous have 2 different phenotypes. And a common example of that is the red gene in snapdragons. So a homozygous red snapdragon is the red flower. There's a mutant which fails to make the enzyme, and if you have a homozygote for that, there's no enzyme, the flower is white. But the heterozygote is not red, as if red was dominant, it's actually pink.
The expression of white in pigeons' color is an example of epistasis.
The white pigeons are an epistatic gene, and this is a gene that actually covers color. And the white pigeons that we're talking about that come from birds that are splashed and that have a very dark eye, when the epistatic gene for white is quite concentrated in a pigeon, the result will be a pure white pigeon. This is a wonderful example of epistasis.
Their expression may be normal in the eye, but this particular kind of mutation is causing it not to be active in the feathers. And what the effect of that is to cover the other possible colors that could be encoded by other genes. So if you have red versus blue-black, those colors are covered by the effect of instructing the feather to make no color. So the white gene is epistatic to the second gene that controls the choice between red and blue-black.
We take birds that are colored, that have white feathers on them, and we cross them with each other. And the F1s that have the most white feathers on them, we cross them with each other. And we continue this on for as many generations as it takes to get a pure white.
The white color in the pigeons is an example of polygenic or multiple factor inheritance, since it takes several generations to get the pure white coloration.
The fact that in these pigeon breeding experiments, the original birds have a few white feathers, and then by progressive breeding you can get a more and more white pigeon, suggests that this actually may be caused by more than one gene. And so we call it polygenic inheritance, where each of the genes individually makes a small contribution towards the total phenotype. And you're looking at the average effect of the states of many genes. And only when you've selected the white allele in all of the cases, or most of them, are you going to approach a completely white bird. It's sort of like a slot machine with multiple rollers on it. Each one will have a little effect, but you really don't hit the jackpot till you get them all saying cherry. All right.
By the time Dr. Carpenter reaches the F3 or third generation, the results are nearly pure white.
In the F3 generation, we get birds that are about, 7/8 white, and we have a few colored feathers. All right, we take birds like this, and we cross them with each other, and in the next generation, we have birds that are pure white.
Breeding animals and birds is a labor of trial and error, one based on patient observation of the laws of inheritance. Dr. Carpenter sees his flock as a labor of love.
Their aesthetic beauty is phenomenal. They're a wonderful genetic animal to work with. Things have progressed where we've been fortunate to make money out of something we love. And what could be better for anyone on the face of the earth to do that?
We all come into this world with genes inherited from our parents. Sometimes, unfortunately, this includes a specific mutation that could cause a life-threatening disorder. These disorders are often linked to the sex of the child and run through a family history.
Well, I was diagnosed at 5 or 6 months old, and the reason my parents were surprised, even though hemophilia was prevalent in my family, is I have 2 older brothers, neither of which have hemophilia. Then I came along and my mother says, "Hey, we must not have hemophilia on this side here. Let's go for another kid." And bada bing, bada boom, the dice came up with bar X. I've had uncles and cousins that had hemophilia, and my mom's mom was a Stuart, going back to the Mary Queen of Scots Stuarts.
Known as the royal disease, hemophilia is visible throughout the family history of the British monarchy, whose offspring intermarried with other royalty across Europe. Charting this royal family tree shows that hemophilia is much more common in males of the family. Such a detailed family history is called a pedigree.
One of the ways of discerning genetic inheritance in man is to look at a family tree. We call these pedigrees. And it is possible to trace in the pedigree the transmission of a particular gene, especially if it produces a disease.
What is hemophilia, and how is it passed to the males of the family?
Well, hemophilia is a bleeding disorder. People who have hemophilia have an abnormal gene, so they cannot make one of the clotting factors that is normally found in the plasma. There are several clotting factors in the plasma. They all have to be there because they form a chain reaction. And if one link of the chain is missing, then the reaction cannot go forward and a clot cannot be formed.
What would be a bruise to you, a bruise for you is a bleeding internally. Well, where yours may stop at a dime-sized bruise, ours keeps flowing and flowing until it fills up a joint area and causes pressure on nerves, which causes pain, stiffness, swelling, and can eventually deteriorate the joint so you'll wind up with arthritis and other difficulties due to the bleedings you have. So it's internal bleedings that we are more concerned with than external.
The clotting factors that are most often deficient are factor 8 and factor 9. And the genes that code for these clotting factors are on the X chromosome.
The location of these genes provides a clue as to why hemophilia is so much more common in males than females. The answer is contained in how the sex-determining chromosomes combine during conception.
Of the 23 pairs of human chromosomes, only one determines a person's gender. In females, this pair is composed of 2 X chromosomes. In males, the pair is an X and a Y chromosome. At conception, the mother's egg contributes an X, where the father's sperm can contribute an X or a Y chromosome. When a zygote receives the X from the mother and the X from the father, the child will be a girl. If the zygote receives the Y from the father, it will develop as a male. In this way, each fertilization has an equal chance of producing a boy or a girl.
Friday evening.
30% of all hemophiliacs show the disease because of a genetic accident called a mutation. The others, like Brian Kraft, can point to a family history. Just how is this gene inherited?
A woman who is a carrier has a defective gene on one of her X chromosomes. So that means that she has the possibility of transmitting that problem to one of her children. If she has a son who inherits the X chromosome with the defective gene, then he has hemophilia. The same way, she can pass on either of the X chromosomes to a daughter. And if it's the chromosome with the defective gene, then that daughter is again a carrier. Whereas if she passes on the chromosome with the normal gene, then that daughter is not a carrier.
When the hemophilia gene is known to be in a family, it's important to understand the ramifications of future inheritance.
The prospect of severe hemophilia should not be taken lightly. There are frequent hemorrhages, frequent intravenous injections.
Over a given year, you can go through $200,000 worth of factor quite easily. And that's just with normal activity and normal care. So it can be quite costly.
The future for hemophilia sufferers will be greatly affected by advances in biotechnology, perhaps even gene therapy.
Gene therapy seems like it's kind of the wave of the future that shows great possibility. And before that happens, I think we'll see totally manufactured clotting factors that have no human products in them at all, which then takes away the risks of any viruses or other contaminants that could be passed along by using the blood supply.
There's a lot of work being done now on gene therapy, but it won't change them fundamentally from being hemophiliacs. If they have children, they have the same chance of having children who are affected with hemophilia as if they had never had gene therapy.
Those who do manifest the disease are left to cope, and few seem to cope as well as Brian Kraft and his wife Kelly.
Kelly and I, we travel around the country doing seminars, often for groups of people with hemophilia or parents of kids with hemophilia, which include humor in everything we do. I can't change the fact that I have hemophilia. However, I can change my perspective on what having hemophilia means. So if I am not happy about having hemophilia, that's serving no purpose. If I can find ways to keep having fun no matter what my physicalness is, then I can lead a happy life and just enjoy getting up every day.
Observing and tracking patterns of inheritance may make it possible for future generations to better deal with, perhaps even avoid, defective genetic characteristics passed down from their ancestors. But in many cases, the mystery of how life looks, behaves, and evolves is still locked in the complexities of inheritance. So the more thoroughly we understand these intricate patterns, the better we'll be able to protect and improve the delicate cycles of life which surround us.